The overall goal of this experiment is to assay active ERK levels in vivo in C. elegans gonads.
This method can help answer key questions in the ERK signaling and germ cell field, such as the effects of drug treatments or gene perturbations on the RAS-ERK pathway and thus germ cell development. The main advantage of this technique is that the ERK signal pattern and strength can be quantified in vivo.
Pick 100 to 150 worms at the desired developmental stage and collect them in a 1.5 milliliter microcentrifuge tube containing 100 microliters of M9 buffer. Then, fill the microcentrifuge tube with 900 more microliters of M9 buffer and centrifuge it at 1000 times G for 1 minute at room temperature. Under a dissecting microscope, gently remove 900 microliters of the M9 buffer. Then repeat the wash 2 more times to remove most of the attached bacteria. Next, using a 200 microliter micropipette tip with a widened bore, transfer the worms in 100 microliters of M9 buffer to a flat-bottom glass watch dish. Then, add 1 to 3 microliters of 0.1 molar levamisole to the dish and swirl it gently. Now, attach 2 25-gauge needles to 2 syringes to make 1 dissecting tool for each hand. Under a dissecting microscope, position each needle under and over each worm and make a fine cut on each worm near the second pharyngeal bulb using a scissor-like motion. Cut all